Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about regulation of transcription. We mainly are going to talk about regulation of transcription, initiation portion of the transcription. Okay. In the previous video lecture we talked about regulation of gene expression in eukaryotes. And before that video lecture we talked about regulation of gene expression in prokaryote as well. We also talked regulation of gene expression as well as level of regulation of gene expression. If you miss this video lecture, so it's very important that first you guys need to watch that video lecture to clear your concept, by which your concept will be clear. Once you guys get idea about these videos lecture, so then come to that video and then this one video will be very easy for you. Now today in this video lecture we are going to talk about regulation of transcription. And all of you better note that in transcription we have initiation, elongation, termination. So in this video we are going to talk about regulation of transcription, initiation portion of the transcription. We are going to talk about that, how the initiation portion of the transcription are regulated. So please stay tuned and keep watching. Okay friends, so now let me write. Okay. Now what does it mean, regulation of transcription? Means that how we can regulate the transcription process. We are mainly going to talk about regulation of transcription in case of prokaryotes. Okay, this is actually in case of prokaryotes. Because in case of prokaryotes the genetic material inside a cell are in a very simple form. And one thing more, that the cell which have a genetic material, we call that DNA have a very linear or in a circular form in case of prokaryotes. But if we talked about, if you guys remember in eukaryotic genetic regulation of genetic expression, which we talked in the previous video, it's very complicated and still we didn't complete it. Still it's very complicated, still a lot of things are remaining and it's still not understandable. That's why regulation of transcription in case of prokaryote are very simple and understandable, and it's very simple process, because in this case the DNA are very simple in a linear circular in form, but in case of eukaryote DNA are very compact in form and a very complicated, the DNA have very complicated structure. Now how the regulation of transcription will occur? Before explaining this topic, first of all, let me a little bit explain the major and basic terms which are very important. Regulation of transcription, in this case, that we are just going to explain that how the transcription process is regulated. Again, if we want to regulate or control the transcription process, still we need different types of protein, different types of enzyme, different types of genes. Okay, so these genes and these types of proteins we are going to talk in this video. Okay, now to explain this process we need to know about the induction and repression of enzyme synthesis. Again, we need to know about that repression and induction of enzyme, because in this case now to understand the transcription in a regulation of transcription, so we need to know about the induction and repression enzyme and their biosynthesis, that how actually it is synthesized. So remember guys, inside the cell what's actually going on inside the cell, different types of chemical reaction are going on and different types of chemical reaction are catalyzed. So these types of chemical reaction are actually catalyzed by different types of proteins. So whenever we have different type of proteins or enzymes, different types of enzymes which are present inside a cell and performing their different functions, so the process through which the enzymes or protein are activating some genes, we call that induction process, and the genes are called inducible genes, while the processes the process through which the enzymes are involved, we call that induction enzymes. And repression of enzyme means that whenever the genes are expressing and we want to stop the expression of that gene. So in this case we need repression of enzyme. So for the repression of enzyme we have repression genes inside the DNA. Okay, one thing more, that inside the DNA what we have a lot of processes carry on. So when these processes, means when different types of chemical reaction which are carry on simultaneously, so then what happen, different types of protein and enzymes are formed. Now remember guys, when the chemical reaction that are going on inside the cell through different types of the proteins and enzyme, we call that a housekeeping function. Housekeeping function. So again, housekeeping function inside the cell, if different chemical reaction are going on and different chemical reaction are catalyzed as metabolic reaction, catabolic reaction, and due to these reaction are actually possible by different types of protein, and these protein performing their different particular type of function, we call that housekeeping function. Because in this case the cell is actually acting as a house. If the protein which are involved in housekeeping function, these protein are actually obtained from a specific genes, we call that housekeeping genes. It means that we have genes available to perform the housekeeping function. We these particular type of genes can producing different types of the protein and enzymes, because inside the cell different processes and reactions are going on to produce different types of the protein. So that's why we have genes, we call that housekeeping genes, that produce protein and perform the housekeeping function. Moreover, this housekeeping genes, these housekeeping genes are expressed continuously. Why? Because different chemical reaction inside the cell are going on at simultaneously time. So that's why these housekeeping genes are continuously expressed. So remember guys, when the housekeeping genes are continuously expressed, then we call that constitutive genes. Okay, so constitutive genes means those genes which are expressed at particular time, or which are expressed continuously, we call it constitutive genes. This process doesn't mean that all the chemical reaction and all the processes inside the cell are going on, means all the genes inside the cell are turned on and all the proteins and enzymes are produced. It doesn't mean like that. It also means that when the cell need, let's suppose if the cell need a beta-galactosidase enzyme, so at that time the cell will turn on their genes which we call a beta-galactosidase genes, means lac genes, they will turn on that gene, and then we get the beta-galactosidase enzyme through this gene. Okay, if we doesn't need, so then that gene will be not activated. Okay, so remember, inside the cell, if a particular gene is required for the particular product, to obtain the particular product, so that gene will be activated at that time. If the cell doesn't need the protein or enzyme, so the genes will be not activated. This is very important question. So now let's suppose we have an enzyme which we already talked in the previous video. Inshallah, in the upcoming video we also explain that enzyme. We called the enzyme beta-galactosidase. Beta-galactosidase. This beta-galactosidase is actually the enzyme which are produced by a genes which we call that lac genes. Lac genes. And these lac genes are actually present in lac operon. Inshallah, we will explain lac operon, trap operon. We will explain the operon concept in detail in upcoming videos lecture. But just here consider that we have a lac genes which produce beta-galactosidase. This beta-galactosidase, what it can do, it will break down the lactose So lactose will be break down into two parts. One we call that glucose, another we call that galactose. Okay, this lactose is actually what this lactose is actually. This is the carbon source, and this is actually the carbon source, and the bacterial cell doesn't need this lactose, but bacterial cell need this glucose. Now how the bacterial cell obtain this glucose? Now let's suppose we have a media. Let's suppose inside a media what we have again the lactose molecule present, but the glucose are not present, and the bacterial cell need glucose. So for that purpose we have a genes. Let's suppose this is a DNA. As you can see, inside this DNA we have promoter region, we have operator region, and these are the structural genes, let's suppose, available at that point. And the structural genes are, let's suppose, called as lac genes, like A, like B, or like Z, X. We will explain it inshallah in upcoming video lecture. And let's suppose this is the DNA. So the bacterial cell doesn't need the lactose, bacterial cell just need glucose, but in a media lactose is present. What actually going on? Inside the bacterial cell, the genetic material are rearranged themselves, and then lac genes will activate it. Now how? Because in this case, this beta-galactosidase, this lactose is actually acting as an enzyme. They will bind to this promoter region to activate the this gene, this DNA, to activate this genes. Let's suppose they activate these genes. So when the genes are activated, transcription process will occur. Okay, and we get mRNA, and from this mRNA we get lac proteins. And this lac protein is actually what? This lac protein is actually beta-galactosidase. Okay. One thing more, this mRNA is actually, in a bacterial cell, the mRNA, when we have one mRNA and it can produce multiple of the protein, we call that polycistronic mRNA. Okay, so the bacterial cell have polycistronic activity because it can produce multiple of protein from this one particular mRNA. That's why in this case not only lac protein are produced, strep protein are also produced, lux are also produced, rex are also produced. So these are actually the protein which are produced from one particular mRNA. We call it polycistronic mRNA. Okay, now if the beta-galactosidase are produced, so then they will break down, cause this lactose is actually disaccharide, so they will cause the breakdown of the lactose and we get glucose. Now the cell will use glucose as energy, and this is how the process is going on. But remember guys, if the glucose inside a media is already present, so then what happened? This process, let's suppose, is activated. We don't need this process. Why? Because glucose is already present. We need this process at that case when the lactose is present. So lactose is present, then beta-galactosidase will produce and it will break down the lactose into glucose and galactose. If the glucose is already present, so then we do not want to switch on or keeping switch on that genes. What happened? At that time, the glucose which is present in a media, it will bind to this promoter region or to this operator region and cause to switch off these genes. So in this case, this glucose is acting as a represser protein or represser molecule. It will bind to the operator region and cause the obstruction of these genes. So these genes will be not activated, and thus this gene will be blocked and we didn't get beta-galactosidase. Okay, this is the process that how it's going on. Let me explain the whole process with an example. Let's suppose inside our house, if we have food, let's suppose we have 1 month food available. If 1 month food is available in our house, so is it mean that we will use all the food in just one day? No, it doesn't mean that. We will use that food which we need right now. So we will use that food. Just like in this case, the bacteria which If let's suppose the bacteria need beta-galactosidase, so at that time the lac genes will activate it. If the bacteria doesn't need, so that gene will be switched off. Okay. Now remember guys, inside a bacterial cell, what's actually going on? Inside a bacterial cell, there are different types of beta-galactose, there are many types of beta, means 3,000 types or 30,000 beta-galactosidase enzymes are available. But if the lactose are present, so then the bacteria will activate that gene again to produce more and more beta-galactosidase. So we also say that this beta-galactosidase is actually acting as an inducible enzyme. This beta-galactosidase acting as an inducible enzyme. Now why we call that inducible enzyme? Because they can induce, they can enhance, they can increase the process of production of beta-galactosidase. Why? Because we have lactose that need to be break down. So in this case, we also say that beta-galactosidase is acting as an inducible enzyme. But this inducible enzyme need activation. This inducible enzyme need activation, and this activation are actually given by an effector molecule, which we call that inducer. So inducer are nothing, these are actually the effector molecules that act upon on the inducible enzymes, due to which the inducible enzyme are activated and bind on the lac genes to activate it and to produce more and more beta-galactosidase enzyme. Okay, so then again another question arises in our mind, that how this inducible genes are produced. How these inducible enzymes are produced. These inducible enzymes are also produced by a genes we call that inducible genes. Inducible. Okay, so this is how the whole process is going on and carry on. Okay, now let me, another thing that's very important, if the cell have enough or let's say if inside the medium the glucose is present in a large number, then this process need to be switched off. Then what happen? We have another molecule which we call it a represser. A represser molecule, what actually it will do, it will cause means we have an enzyme which we call it represser enzymes. Let's suppose we have, means this is repression enzyme. This repression enzyme will not in an active form. It will present inside a cell, but it is not in an active form. For the activation, it need a co-repressor molecule. Now it will bind with the repressor enzyme. So when it will bind, this co-repressor are bind. So then it will this repressor enzyme will activate it. The repression enzyme will activate it, and it will bind to this operator region. So when it bind to this operator region, so the process will be stopped. So when the process stopped, so then we what happened? Beta-galactosidase or the proteins not produced. Okay, so we call that a repressor enzyme. Okay, and these repressor enzymes are also produced by a genes we call that repressor genes. So that's all about the regulation of transcription initiation, and that's actually what we talked. We talked the induction and repression of enzyme synthesis. So we actually just talked the most important terms and most important genes, as well as we talked the basic idea about the regulation of transcription. Okay, this is not the whole process. Inshallah, in the next video lecture, we will explain that how the transcription of initiation will be regulated by a regulated protein. Means how we can, regulate the transcription process by a regulatory protein. There are some other regulatory protein which are mainly involved in the regulation of transcription initiation. Okay, I hope you guys get idea about that. If you like this video, make sure to hit the like button, share this video, and subscribe the channel to get more interesting videos like that. Thank you so much.